Hello, and welcome to this week's episode of Medisodes. This week, we're going to be discussing another fundamental system of the human body, the immune system. The immune system helps get rid of any foreign and potentially disease-causing pathogens that may enter a person's body. There are several parts to this complex system. The immune system is divided into this non-specific and specific responses, with a non-specific response covering inflammation, phagocytosis, and antimicrobial proteins, and the specific response consisting of B cells and T cells. To start off today's episode, I'm going to hand over to Shrey, who will be talking about the non-specific immune system.
So, the non-specific immune system is the body's immediate immune response to the detection of an antigen from a pathogen. And as the name suggests, it's not targeted at a specific type of pathogen, or it's more looking at trying to fight this threat that the body perceives as soon as possible with as much as it can do. So let's start with lysozymes. The lysozymes are enzymes that kill the bacteria or other pathogens by breaking down their cell wall. And they break down the cell wall by hydrolyzing the polysaccharide chains that are found within bacteria cell walls. And they can be found in tears, in saliva, and in nasal secretions. So really around where the openings of your body can be, where you're susceptible to infection. And they play a really important role. Next is fevers. So, as you may know, the hypothalamus regulates the normal circadian rhythms of body temperature. However, when the body's infected, the hypothalamus resets itself at a higher temperature. So if you've learned about homeostasis, you'll know that there's a norm value. When you're infected, this norm value is increased. And what this aims to do is to reduce the ability of pathogens to reproduce efficiently, because they're not at their optimum temperature, and some of the enzymes that they use might be denatured. And this is also predicated on the fact that the human body can survive higher temperatures than those of bacteria. Otherwise, this fever method wouldn't work. Also, the specific immune system is adapted to work better at higher temperatures. So this has a twofold benefit. And many of you, when you've been sick, will have these fevers. And if you've listened to our episode on the common cold, you'll also know that fevers are really important. Next, inflammation. So when you have an injury, that injury and that maybe the breaking of the skin allows microbes and pathogens to get into the body. So firstly, your wound is sealed by a blood clot made of platelets, and that's really important to stop further infection. And it's really important also to be cleaning the wound to stop to kill any microbes that might try to enter the body. But after that, you will have an inflammatory response, which helps to destroy all the invading microbes at the site of the wound. So what it's made up of is damaged white blood cells and mast cells in the connective tissue below the skin, and around the blood vessels will release histamine. And what this histamine does is it causes local arterioles to dilate, and this increases the blood flow to that region where the wound is. And there's also increased permeability of capillaries. The pores in the capillaries will get wider, and which causes them to leak. And plasma fluid, white blood cells, and antibodies can then leak from the blood into tissues, and this can form an edema. The remaining microbes that don't get swept up by these things are then attacked by new intact white blood cells brought about by the increased blood flow. But by far the most important process in the non-specific immune system is phagocytosis. So phagocytes are a type of white blood cell that engulf bacteria and other foreign matter in the blood and tissue. So pathogens and damaged cells will release chemicals, and these will attract the phagocytes to the area where there is infection. There are two main types of phagocytes. Those are neutrophils and macrophages. And how they work is engulfing the pathogens and then digesting them. So neutrophils can engulf about 5 to 20 bacteria each, Macrophages can destroy up to 100, um, but you have to remember that there are many thousands, even millions of these. So, but there's also millions of pathogens. So it's a constant battle of numbers. And if phagocytosis is successful, within a few days, the infected area um, should be it will be full of dead cells basically, and this can form pus, which is obviously not very nice, but it's better than having an infection. So if we look at how phagocytosis actually works, it's that the antigen or the marker from that bacteria will be engulfed by a neutrophil or a macrophage, and this is then enclosed in a vacuole, which is kind of like a little sealed fat blob inside the cell. From there, lysosomes, not to be confused with lysozymes, which are basically these packets of um, digestive enzymes, will fuse with this vacuole and they'll release the enzymes, and that destroys the foreign material. But a couple of these pathogen antigens are left, and these combine with special proteins that form an antigen protein complex. And these antigen protein complexes are displayed on the outside of the macrophage and basically acts as a messenger to other macrophages to tell them that there's an infection and what is causing the infection. Neutrophils actually produce these things called radicals. So you might have learned it from chemistry. It's a substance with an unpaired electron. And for example, these can be nitric oxide and superoxide. And what these radicals do is they attack the bacteria's DNA and their protein. However, excess radicals also destroy human DNA and human enzymes, which is obviously not very good. And if you've ever heard of being told to eat antioxidants, that's what antioxidants do. They get rid of these radicals to stop them damaging our body. So macrophages and lymphocytes are found in the lymph nodes, the liver, and the spleen, and this helps them to hinder spread and have a rapid response. Also, and it's really important that this system works, because if the system fails, there can be widespread infection, septic shock, and blood poisoning, which has very high chance of fatality. The last part of the non-specific immune system I'm going to talk about are antimicrobial proteins, otherwise known as interferons. So what interferons do is they target viruses specifically. So most of the things we've been talking about so far have mostly been targeting bacteria, but this is specifically for viruses. And interferons are produced by virus-infected cells, and they bind to the cell surface receptors of uninfected cells. And they basically tell the cells that this virus is coming, and they prevent viral replication by inhibiting the viral protein synthesis enzymes. And therefore, the viruses cannot replicate and carry on in the lytic cycle. And that helps to stop the spread of infection so quickly. It was hoped that actually that interferons would help produce antivirals. However, they're only produced in tiny amounts in the body, not enough to be used therapeutically. However, with new techniques such as genetically modified bacteria that can produce commercial quantities of the interferons, even if they're very expensive, we might be able to see uses. For example, interferon alpha is used to treat hepatitis C. There are some, as with any new treatment, there are side effects, and we don't exactly know how effective they are yet. But it could be very promising in terms of using interferons to treat viral infections, especially in the context of, for example, COVID-19. So we've talked about the non-specific immune system, and it plays a really important role in fighting infection. But now we must move on to the next stage to talk about the primary specific immune system with Adrian.
So, as Shrey mentioned, the immune system is not limited to just these non-specific responses. The non-specific immune system targets all foreign pathogens, but this is not as efficient as a specific immune system, which targets one specific pathogen. Antigens are molecules that are present on the surface of all cells. Every cell has specific antigens. This allows the body to identify them. Cells that are identified as self are those that are their own body cells, whereas cells that are identified as foreign induce an immune response. Lymphocytes are a type of white blood cell that help to defend the body against specific diseases. They circulate in the body and lymph fluid, and gather in large numbers at the site of any infection. Reserve supplies of lymphocytes are held in strategically positioned lymphoid tissue. There are two main types of lymphocyte, B cells and T cells. Both types respond to antigens, proteins, and other chemicals that are identified as foreign and are not normally found in the body. B cells are produced in the bone marrow. Each B cell has one type of antigen receptor on its surface, and is activated when it binds to an antigen with its complementary shape. Once activated, the B cell secretes antibodies in response to the antigen. Antibodies are specific protein molecules that bind to the antigen on the membrane and act as labels to allow phagocytes to quickly identify and destroy them. T lymphocytes are also produced in the bone marrow, but unlike B lymphocytes, they mature in the thymus gland. They exist in the lymph nodes, ready to respond to any passing pathogen. There are two types of T cell, T helper cell and T killer cell. The specific immune response is split into two distinct stages, primary and secondary. The first part of the primary immune response is the activation of T helper cells. When a macrophage engulfs a pathogen by phagocytosis, it displays the foreign antigen on its own surface and becomes an antigen-presenting cell, or APC. A T helper cell with complementary shaped receptors, called CD4 receptors, on its surface, binds to the antigen on the surface of the antigen-presenting cells. Once activated by this binding, each T helper cell divides to produce a clone of active T helper cells and a clone of T memory cells. The next part of the primary immune response is a cloning of B cells. A pathogen with the foreign antigens binds to a B cell with a complementary receptor, and it becomes a B cell APC. The activated T helper cell from the previous stage. The activated T helper cell from the previous part produces cytokines, which stimulate the B cell. This B cell then divides into B memory and B effector cell. The B effector cells are differentiated to produce plasma cells, which secrete antibodies specific to the antigen. Finally, T killer cells are also involved in the specific immune response. If a bacterium or a virus invades a body cell, then it is located within that cell and is not accessible to the rest of the immune system, which acts outside the body cells. Once inside the cell will present the bacterium's or virus's antigens on its surface and becomes an APC, an antigen-presenting cell, much like a macrophage would. A T killer cell with a complementary receptor to bind to the antigen presented on the body cell, and this stimulates them, cloning to produce T killer memory and T killer active cells. These T killer active cells then spread throughout the blood and bind to any body cells that are presenting antigens on its surface. These T cells then release chemicals by These T killer cells then release chemicals by exocytosis, which causes pores to form in the cell surface membrane, causing cell lysis, which means the cell bursts and dies. This also kills the pathogen in the process, and thus is a way of fighting this infection. I will now hand over to Anupam to talk about the specific immune response, and to go through a specific example of an immune response in tuberculosis.
The secondary immune response occurs only if a person is infected by the same bacterium or virus again. This is because the secondary immune response relies on memory cells that are produced during either the role of T killer cells or during the cloning and activation of T helper cells. The immune system responds much faster in the secondary immune response and can take about 2 to 7 days. In most cases, the person will not even realize that they've been infected, as the immune system is that efficient in destroying the pathogen. B memory cells, which were produced in the primary response, differentiate immediately to produce plasma cells and release antibodies. This production of antibodies is far greater, and the response lasts longer. This means that the invading viruses and bacteria are destroyed rapidly. At this point, a person is said to be immune to a disease, as they will not feel the side effects of having that disease. It's important to note that after a secondary immune response, while the levels of present memory cells in the body can decrease, this takes sometimes many months and many years for that immunity to go away. And this immunity can also be artificially generated by being exposed to a vaccine, where the memory cells need not be present, and instead the body thinks that it is being attacked by a pathogen, but instead it's just being trained so that when a real infection comes, it can fight it that much more effectively. As an example of how the immune response system works, let's talk about tuberculosis. Tuberculosis, or TB, also known as consumption, white plague, is a contagious disease caused by a bacterium called Mycobacterium tuberculosis. It's an ancient disease, but that doesn't mean it's still prevalent in modern day society. There's estimated to be about 6 million new cases each year, and 1.5 million people die every year from the disease. The primary infection of TB can often last for several months and may not have any symptoms. The immune system of a person infected with TB responds immediately to deal with the infection. The M. tuberculosis bacterium inside the body causes an inflammatory response, as Schrey described in the nonspecific immune system, as well as having macrophages engulf the bacteria. This can cause masses of tissue known as granulomas to form in response to infection. And in TB, these tissue masses are anaerobic and contain both dead bacteria and dead macrophages inside them. These are called tubercules and give the disease its name. And after a period of 3 to 8 weeks, the primary infection is controlled and the infected region of the lung heals. Most primary infection happens during people's early childhood, and 90% of infections heal without ever being noticed. The part that makes TB so dangerous is that the bacteria evade complete destruction by the immune system, as while normally macrophages would engulf and destroy bacteria, M. tuberculosis bacteria can actually survive inside the macrophages. After they're taken up by phagocytosis, they resist the killing mechanisms used by the cells, such as digestive enzymes. This is due to their thick, waxy cell walls, making them very difficult to break down. And this can cause live but inactive bacteria to remain with the tubercules and granulomas that form for many years. While the immune system controls the infection, preventing the spread of bacteria, it is always present in their body, ready to spread at a moment's notice. TB bacteria can also suppress T cells, reducing the antibody production and attack by T killer cells, making it that much more lethal when it enters the second phase, active tuberculosis. This occurs if the patient's immune system cannot contain the disease when it arrives in the lungs or after it breaks out of the tubercules. This may be because the number of bacteria is too great, or alternatively, it could be because the immune system is no longer working properly or is suppressed in some way. About 80% of active TB cases are caused by reactivations of previously controlled infections. With active tuberculosis, the bacteria multiply rapidly and destroy lung tissue, creating holes or cavities, eventually killing the sufferer if they're not treated with an appropriate antibiotic. Nowadays, however, most people in parts of the developed and developing world will be vaccinated against tuberculosis. The vaccine introduces the person's immune system to a safe, attenuated version of the bacterium before they are ever infected with TB. And this means that their body's immune system already has the capabilities to ensure that the bacterium is destroyed upon first contact, preventing the formation of tubercules and preventing reactivation later down in their life. Hopefully that example gives you a deeper insight into how this immune system works and why it's so effective at keeping all different types of diseases, whether they be pathogens in the form of viruses, bacteria, or other types of foreign debris inside the body, at bay, and making sure the human body stays healthy for years after even the infection. For a summary, I'm going to pass over back to Adrian.
So that covers the majority of the immune system, including both specific and non-specific parts. And the example with TB really showed how all of it comes together to protect the body. Thank you for listening to this week's episode of Medisodes. Be sure to like and subscribe, and tune in for next week for another episode.